Speak Student!
Molecular genetics, DNA extraction, a la Shmoop. Okay, we're back with you in the land of biology, and we're going to talk about DNA extraction with Dr. Ruth Tennant here, who is a guru on the subject. So, Dr. Ruth, why would anyone want to extract DNA in the first place? Like, what does that do for us? Like, when I go to Vermont, I understand why I want to extract maple syrup, because it tastes good. Why would anyone want to extract DNA?
Similar idea. So maple syrup doesn't do you any good when it's in the tree. Just
Right.
like if you want to study the DNA, you need to get it out of the cell. So tons of reasons you might want to do this. Let's say you want to do some forensic testing. So you have found DNA at a crime scene and you have a suspect. You got to take his DNA. You have to extract his or her DNA out of their cells and test it. Same thing with genetic testing. You can't really sequence or figure out what the sequence of DNA is while it's in someone's cells. So you have to burst open the cells, take out the DNA so that you can study it and sequence it.
Wow, that's really cool. Okay, so the obvious question is, how do you extract DNA? And like, can we buy a kit that does that for home use?
You can buy a kit if you're in the lab. At home use, it's actually super easy, just using kitchen ingredients. There's just a few steps. So the first thing you have to do is break open the cells, and you can do that by grinding them. You can often add detergent. So soap is something that will break up basically.
Wait, go back, grinding cells.
Yeah.
I'm thinking about like the maize flour
The
things
mortar
you
and
take.
pestle, yeah, that'll do it.
Wow.
Yeah.
So wait a sec. So we've got dried blood from the guy who got shot, and you like stick it in a bowl, and it dries, and you pound it.
So probably if they were doing it in the lab, they would not use that. They have like a little tube
Right,
that'll do
understood.
everything. But like if you want to take your cheek cells, you basically could just like slough them off using Gatorade and then spit them into a tube, and they would kind of that sort of mechanical chewing would. start
Wow,
breaking them
that's
open.
gross and really cool at the same time.
Pretty awesome.
Yeah. Okay, keep going.
Sure. So you got your cells that you got out of your organism, however you did, and then you would add detergent and salt, which basically breaks open the cell membrane and the nuclear membrane. So that'll kind of release the DNA. The problem at that point is that the DNA is still mixed up with all the other gunk in the cells.
Got it. When you say release the membrane, so the membrane's like the skin around it, like a balloon or something, and that melts the skin, and the goop inside then kind of comes out.
Exactly. So the membrane is made up of phospholipids, so like fats, and so if you add detergent to that, it'll break it up just like it'll break up grease on a plate.
Got it. Okay. So then we've added rubbing alcohol.
So that's the next step, exactly.
Okay.
So you've got your DNA, but it's mixed in with all this other stuff in the cell that you don't want. And DNA has an interesting property where if you add rubbing alcohol or even ethanol, it'll precipitate out of solution, which means that it will form a solid when it used to be in solution.
And that's because like the alcohol evaporates quicker or something, and what's left is.
The way that I usually think about it is that DNA likes to dissolve in water, but it doesn't like to dissolve in alcohol, so it's kind of like scared out of the water, and when it hits the interface between the water and the alcohol, it kind of forms just a solid right there.
That's how Michael Phelps did it. So then go through then. So what happens? Then the alcohol dries, and we stick the goopy stuff back in the water.
So yeah, what you'd end up with is you'd have like a little pellet of dried up DNA, which is also not useful because you ultimately need it to be in a liquid form for most of the tests that you do. So you just add some water, wait a little bit, heat it up, and then it redissolves, and you've got pure DNA in liquid form. Woo!
Wow, that's really cool.
It's pretty cool, and it's real easy, especially if you do it with something like strawberries that have tons of DNA.
All right, well, kids, do try this at home. Amaze and amuse your friends. Okay, that's it, DNA extraction. Why would anyone want to extract DNA? How do you extract DNA?